Good afternoon, everyone. My name is Melinda Hunter, and today, along with Megan Barnum and Eric Leday, we'll be presenting our experiment, which is the effects of concentration of silver nanoparticles on crawfish in a clay and water habitat. The purpose of this project was to monitor varying levels of silver nanoparticles and the effects that they have on the behavior and death rate of crawfish. We all know that Louisiana crawfish is an indigenous food staple here, and in the industry it produces billions of dollars. However, silver nanoparticles are also invading their habitats through point source and non-point source pollution from chemical plants to farms and varying water sources. So, what we decided to do was to take this xyro specimen and measure the amounts of silver nanoparticles that we can put in the water before the crawfish reach their LD50, which is the live death rate at which 50% of them have died. We all know silver nanoparticles are actually highly neurotoxic to freshwater vertebrates. So they specifically affect their nervous system. And in order to do this, we wanted to measure the microscopy of those organisms. So what we did was we created a pre-treatment habitat for the crawfish. As you can see in the picture above, in the habitat we use netting to separate them, and we also use pond water, deionized water, and rocks to keep the crawfish in the tank because they had a tendency to crawl out. They were all kept under the same conditions for 48 hours before we began our experiments. The pre-treatment tank design. As you can see, the tank was consistent of a tub that used netting to separate the crawfish, and then we placed the netting on top and used rocks to hold it down. The tanks for the testing were actually 4 separate tanks. We have 1 control tank and 3 variable tanks, which were tanks 2 through 4 and tanks 1 was our control tank. For the crawfish, we had to go to the Aquaculture Research Center and sort and specify which kind of crawfish we wanted. We wanted all the crawfish to have similar body masses as well as be around the same weight and length, which was about 3 inches. For the water, we used 2 liters of deionized water. We used 2 liters of pond water collected from the Aquaculture Center, and then we also aerated using oxygen pumps located within the aquaculture lab. For the materials, with the silver nanoparticles, we got them in separate batches of 700 parts per million, 2,000 parts per million, 4,000 parts per million, and then we also collected 10 kilograms of clay for each tank. For the methods, in order to measure the levels of SMP, we manually stirred in the silver nanoparticles into the tanks, and then we sonicated it to make sure they were evenly dispersed. Then we mixed in each of the 10 grams of clay, and then we placed the crawfish in. We measured the crawfish for approximately 72 hours at 3-hour intervals, with each group member going to the aquaculture lab to look at the crawfish. And once the LD50 was determined, the experiment was stopped. We then put all of our data and results into a chart and table, and we found that after about 48 hours, 50% of the crawfish died in the 7,000 parts per million tank, showing that crawfish do not have a high level of immunology to the silver nanoparticles. For our future work, we would like to reconduct this experiment using a longer time interval, as well as dividing the time interval into smaller periods of checking the crawfish. We also want to use a specific type of clay. The clay we used was actually found within the wetlands here in Louisiana. So what we're looking for is to use a bentonite, a kaolinite, or westernite clay. We also want to use further measures to measure the causes of the death rate within the crawfish, whether that be through microscopy and also dissecting crawfish to look at their gills. If there are any questions or comments, I'll take those now. Thank you.